Assalamu alaikum. Today we want to talk about chapter 17, glycolysis. Now in the previous chapters we talked about the structure and function of biomolecules like peptide and proteins, lipids and carbohydrates. Now we will spend the next 4 chapters talking about carbohydrate metabolism because of its central role in life processes. Now the subject of this chapter is glycolysis, the conversion of glucose to pyruvate, and then we will talk about the control of this pathway. Chapter outline. In part 1 we will talk about the overall pathway of glycolysis and the conversion of 6-carbon glucose to 3-carbon glycerol dehydrophosphate, and then glycerol dehydrophosphate is converted to pyruvate. Why glycolysis? Because it is the first stage of metabolism from bacteria to humans, where 1 molecule of glucose is converted to 2 molecules of pyruvate and passing through the formation of a compound called fructose 1,6-bisphosphate. Now, of course, this play a role in the way organisms take energy from nutrients. Now, the glycolytic pathway, also known as the Meyerhoff Pathway. Now, it consists of 2 phases. Phase 1 is the energy requiring phase, where 2 molecules of ATP are consumed. And in the second phase, called energy yielding phase, where 4 molecules of ATP are produced, giving a net gain of 2 ATP molecules for each molecule of a glucose that passes through glycolysis. Now, once pyruvate is formed, it has one of several fates. Now, in the presence of oxygen, pyruvate here loses CO2 and the 2 carbons are attached to coenzyme A to form acetyl-CoA, and this will enter into the citric acid cycle. And this is aerobic oxidation followed by oxidative phosphorylation to form CO2 water with a number of ATP, and we will talk about this in the coming chapters. Now, in the absence of oxygen, however, pyruvate undergoes the so-called anaerobic glycolysis, and this occurs in the muscle. This is, by the way, the more common fate of pyruvate is its reduction to lactate in the muscle, whereas in some organisms like yeast, which undergo the so-called alcoholic fermentation, pyruvate is converted first into acetaldehyde and then into ethanol plus CO2. Now notice here that the 2-NADH, the reducing equivalents that were produced in glycolysis, are consumed, however, in anaerobic pathways in muscle and in yeast. What is the importance of anaerobic metabolism? It's the only source of energy or energy source for red blood cells, as well as some species of bacteria called lactobacillus in sour milk and Clostridium botulinum in canned foods. So this is the importance. Now, in all of these reactions, the conversion of glucose to a product is an oxidation reaction, which means it requires an accompanying reduction reaction in which NAD plus here, this is the oxidizing agent, is reduced to NADH. So this figure 17.3 shows the reaction sequence with the names of the compounds. And note that all sugars in this pathway have the D configuration. And here you can see the anaerobic fates of pyruvate plus the aerobic fate of pyruvate. Here we have the aerobic, and these two, one in muscle and one in yeast. Now, glycolysis consists of 10 reactions we begin with phosphorylation of glucose to give glucose 6-phosphate. Now, glucose can enter and come out of the cell freely, but in order to trap it inside the cell, it becomes phosphorylated because neutral glucose can come and enter, but negatively charged glucose cannot come out of the cell, so it is forced to continue the other 9 reaction. Now the second reaction called isomerization of glucose 6-phosphate to fructose 6-phosphate. Glucose and fructose, both are hexoses, but one glucose is an aldohexose and fructose is a ketohexose. So you need an isomerase enzyme, as we will see. Step 3, phosphorylation of this fructose 6-phosphate to give fructose 1,6-bisphosphate. Then cleavage of this compound to give 2,3 carbon fragments. One is called glycerol dehydrophosphate and the other called dihydroxyacetone phosphate, followed by another isomerization of dihydroxyacetone phosphate to give glycerol dehydrophosphate. Now, this stage is important because now you have, instead of one, you have 2 glycerol dehydrophosphate molecules to continue with. Then, step 6, oxidation of glycerol dehydrophosphate to give 1,3-bisphosphoglycerate. This is the only oxidation reaction in glycolysis where NAD plus, as I said, is reduced to NADH, and NAD plus act as the oxidizing agent. Now, the compound 1,3-bisphosphoglycerate is a high energy compound. Transfer of a phosphate group from 1,3-bisphosphoglycerate to ADP to give 3-phosphoglycerate with the formation of an ATP. Then isomerization of 3-phosphoglycerate to 2-phosphoglycerate. Then dehydration of 2-phosphoglycerate to give phosphoenol pyruvate. And again, this is a high energy compound where in the final reaction, transfer of a phosphate group from phosphoenol pyruvate to ADP to give pyruvate with ATP2. So note that only one of the 10 steps in this pathway involve an electron transfer reaction, and that is reaction number 6. Now we will look closely at each of these reactions in detail. Now again, this shows you the conversion of glucose to glycerol dehydrophosphate, names of intermediates. Notice here, as I said, after this step we have 2 molecules of glycerol dehydrophosphate and they go parallel to each other in the following reactions. Or you can follow one and multiply by 2 anything that is produced or anything that is consumed. Here you can clearly the reaction that consume ATP and the reactions that produce ATP, and also the only oxidation reaction that produces an ADH. So as I said, we have 2 phases. In the first phase of glycolysis, 5 reactions convert a molecule of glucose to 2 molecules of glyceraldehyde 3-phosphate from glucose to glyceraldehyde 3-phosphate. This is phase 1, and the next figure shows you the structure of reaction components here associated with such conversion of glucose into glyceraldehyde 3-phosphate. Notice here we have glucose in the preferred Haworth projection formula, and these are the Fischer projection formula, the one we studied in chapter 16. And here you can see the place of the aldol cleavage. This is aldol cleavage here to produce dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now we will go through each reaction alone, but first let's look at the reactions of the 10 reactions of glycolysis with their standard free energy changes. Notice that here we have 2 values. We have delta G naught and delta G naught prime. Now for biochemical reaction, delta G naught was modified. All reactants and products has 1 molar concentration except hydrogen, it is modified to have a value of 10 to the minus 7 molar so that the pH equals 7, not 0. So delta G naught prime called the modified standard free energy change, and this is especially for biochemical reaction, because from now on any reaction you see has a delta G naught prime value. This could be negative or positive. Negative exergonic reaction and positive intergonic reaction, and you can see these values here. There is the concept of coupling in metabolism where the energy released by exergonic reaction drives endergonic one. The whole process is exergonic. Now let's take the 10 steps step by step. Step number 1, conversion of glucose to glucose 6-phosphate. As you can see here, this is the Haworth reduction formula for glucose. You see the anomeric carbon number 1 here. This is plus ATP in the presence of enzyme called hexokinase. Now the glucose is phosphorylated at carbon 6. So this process of phosphorylation is endergonic, but it is driven how? By the hydrolysis of ATP. We know when ATP, when it's hydrolyzed to ATP, produces minus 7.3 kilocalorie per mole. So this drives the reaction actually. The enzyme hexokinase has an iso form called glucokinase in the liver. So all cells contain hexokinase, but liver cell, they have glucokinase. And it is specific for glucose. Here we talk about the substrate glucose, but hexokinase might use other hexoses such as mannose, galactose. Now a word about the kinase. Now the term kinase is applied to a class of ATP dependent enzymes that transfer a phosphate group from ATP to the substrate, in this case glucose. As I said, we could have another substrate other than glucose. We could have, for example, fructose and mannose. Now the product glucose 6-phosphate inhibits the activity of the enzyme hexokinase, and this is of course a control point in the pathway, as we will see later on, and this is called product inhibition to distinguish it from feedback inhibition. Liver glucokinase needs a much higher substrate level to achieve saturation than hexokinase does. So when glucose levels are high, the liver can metabolize glucose via glycolysis preferentially over the other tissue. When glucose levels are low, then hexokinase is still active in all other tissues. Now, if we, of course, this enzyme also requires the cofactor metal ion Mg+2. Now, if we look at this figure, which shows us a comparison of the conformation of hexokinase before and after substrate binds. Here you see here we have, this is the free enzyme hexokinase, and here we have a cleft for binding of glucose and ATP, and then another cleft which divides molecule into two loops. Now this is the substrate glucose. When glucose approaches and is bound in the cleft, the upper loop here, the upper loop moves down with respect to lower loop to lie over glucose, and this is a good example of the so-called induced fit model for enzyme binding its substrate. This is called induced fit model, and the one we talked about in chapter 6, the induced fit theory. In all kinases for which the structure is shown here, a cleft closes when substrate is bound. Now in step 2, glucose 6-phosphate is isomerized to fructose 6-phosphate, the enzyme called glucose phosphate isomerase. Notice here that carbon 1 of aldehyde group of glucose 6-phosphate is reduced to the hydroxyl group, and carbon 2 hydroxyl group is oxidized to give the ketone group of fructose 6-phosphate with no net oxidation or reduction. As I said, both glucose and fructose are hexoses. One is an aldo hexose and the other is a keto hexose. Step 3, however, step 3, conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. Notice here a second phosphorylation reaction here, where a second mole of ATP is consumed and is the source of the inorganic phosphate that is added to fructose at position 1. So we get by the enzyme is called phosphofructokinase. This is an allosteric enzyme, and the product is called fructose 1,6 bisphosphate. Now the enzyme itself is a tetramer that of two types of subunits, M for muscle and L for liver. When we say M4 and L4, these are homogeneous forms, M4 in the muscle and L4 in the liver, and these heterogeneous forms are found in the blood. So how many? We have 5 isozymes. These are called 5 isozymes, and they can be separated from each other by electrophoresis. In red blood cells, several of the combinations can be found. So these are the possible isozymes of phosphofructokinase. Now this figure shows us the allosteric effect in phosphofructokinase. ATP here is an allosteric effector in the reaction. Here, when ATP is high, the curve is sigmoidal and it inhibits phosphofructokinase. So the rate is depressed. However, low levels of ATP stimulate the reaction. And this is of course reasonable, because when the cell has high ATP concentration, then there is no need to metabolize glucose for energy. So this inhibits the glycolytic pathway itself. In addition to ATP, we have another more potent allosteric effector of the enzyme phosphofructokinase, and this is called fructose 2,6 bisphosphate, and we will discuss this in the next chapter. Now step number 4, however, fructose 1,6 bisphosphate is split into 2,3 carbon fragments. Now this is a cleavage reaction, and this is described as the reverse of an aldol condensation. The enzyme that catalyzes this reaction is called aldolase, and it is isolated from most animal sources, but the one from muscle is extensively studied. The basic side chain of an essential lysine residue plays the key role in catalyzing the reaction. Of course, the thiol group of cysteine also acts as a base. Now reaction number 5, the dihydroxyacetone phosphate is converted to glycerol dehydrophosphate. The enzyme called triose phosphate isomerase. Delta G for this reaction under physiological condition is slightly positive, as you can see, plus 2.4 kilojoule per mole. But as I said before, coupling occurs. So this reaction can be driven by coupling with the reaction before or reaction after, because glycolysis has several reactions that have very negative delta G value. And coupling is what metabolism is all about. Now, this also shows us the conversion of dihydroxyacetone phosphate into glycerol dehydrophosphate by a triose phosphate isomerase. This is the reaction. By the way, all the reactions of glycolysis are reversible except the 3. Step number 1, step number 3, and step number 10. And these are, as we will see later, are control points in glycolysis. Now this is the end of phase 1, where this phase we generated 2 molecules of glycerol dehydrophosphate, and with the consumption of 2 ATP. Now we go to phase number 2, where this glycerol dehydrophosphate will be eventually converted into pyruvate, and it is referred to as the payoff phase since ATP is produced. And again, this figure shows us the places where ATP are being produced or consumed, as well as the point where NADH is produced. Now the following figure shows the molecular structure at each reaction. So this is similar to this, but with the structure. Here we have Fischer projection structure for all intermediates that glycerol dehydrophosphate pass through to form pyruvate. And we will talk about them also in detail by going to step number 6. So, glycerol dehydrophosphate now is oxidized to 1,3-bisphosphoglycerate, and this figure shows us the reaction. Notice here we need an oxidizing agent NAD+. Also we need an inorganic phosphate which is added to the substrate to form this 1,3-bisphosphoglycerate. You might think this is a single reaction. Actually, this is a combination of 2 reactions. The first one is the oxidation of glycerol dehydrophosphate into the oxidation of glycerol dehydrophosphate by NAD+ to form 3-phosphoglycerate. Here notice that the aldehyde is oxidized to the corresponding carboxylic acid. And then the second step is phosphorylation of 3-phosphoglycerate, adding this to this to form 1,3-bisphosphoglycerate. So when you combine these 2 reactions together, you get the net reaction which you saw previously. And again, delta G naught prime can be added for the 2 reactions to get the final delta G naught for the whole reaction. The enzyme that catalyzes this reaction, called glycerol dehydrophosphate dehydrogenase. This enzyme is an NADH-linked dehydrogenase. The structure of this and other dehydrogenases have been studied with X-ray crystallography, and all of them, they have similar structure, are not similar to each other, but the structure of the binding site for NADH is quite similar in all of these enzymes. So what is similar is where NADH binds. The oxidizing agent is NAD+. Here, both oxidized and reduced forms of the coenzyme bind to the enzyme. Here, notice that this is in yellow, the nicotinamide portion of NAD+, and here in red, the adenine dinucleotide portion of NAD+. And here you have the substrate here, and these are sites of interaction. The enzyme itself is a tetramer consisting of 4 identical units. Now each unit binds 1 molecule of NAD+. So actually you need 4 molecules of NAD+, not 1, to accomplish this reaction. Each subunit contains an essential cysteine residue, and this is the only oxidation reaction in glycolysis. Now the product is compound. The product 1,3-bisphosphoglycerate fulfills this requirement for production of ATP since it has a high energy phosphate bond. And as we will see in the next step number, this is the role of active cysteine residue in glycerol dehydrophosphate dehydrogenase. Notice here that this is your substrate and this is the enzyme with the cysteine or the thiol group at the active site. It acts as the sulfur acts as a nucleophile, attacks the carbon to give a tetrahedral intermediate, which goes with rearrangements, etc. And then the inorganic phosphate The oxygen here also acts as a nucleophile. It attacks this carbon to produce a tetrahedral intermediate, which طبعا rearrangement occurs to give the products and to generate the thiol group of cysteine that is on the enzyme. Now, the coming reaction, step number 7, the two reactions in which ATP is produced by phosphorylation of ATP. So this is one of two reactions in which ATP is produced by phosphorylation. Now the enzyme that catalyzes this step is called Phosphoglycerate Kinase. 1,3-bisphosphoglycerate transfers the phosphate group to ADP producing ATP, and again this transfer is typical of so-called substrate level phosphorylation, to be distinguished from oxidative phosphorylation, which we will talk about in chapter 20, in which transfer of a phosphate group is linked to electron transfer reactions in which oxygen is the final electron acceptor, as we will see in chapter 20. The only requirement for substrate level phosphorylation is that the standard free energy of dehydrolysis reaction is more negative than that for hydrolysis of the new phosphate compound being formed. And this is the case here. So reaction is the sum of the endergonic phosphorylation of ADP and the exergonic hydrolysis of the mixed phosphate anhydride. As we can see in this figure, which shows us the dephosphorylation of 1,3-bisphosphoglycerate to 3-phosphoglycerate. And the enzyme, again reversible reaction, which requires Mg+2 as a metal ion cofactor. Actually, all kinases require Mg+2 for their reaction. Step number 8, where 3-phosphoglycerate isomerizes to give 2-phosphoglycerate. Now the enzyme phosphoglyceromutase, as you can see here, catalyzes the isomerization reaction where this phosphate group is transferred from carbon 3 to carbon 2 by this enzyme called phosphoglyceromutase, and it requires Mg+2. A simple isomerization reaction. It is not called isomerase mutase because it transfers a phosphate group, not as we saw between the conversion of glucose to fructose, which required an isomerase. Now the reaction number 9, the conversion of 2-phosphoglycerate into phosphoenol pyruvate by loss of one water molecule. The enzyme is called enolase, which catalyzes this reaction. It requires Mg+2 as a cofactor. Actually, the water molecule that is eliminated in this reaction binds to Mg+2 during the course of the reaction. Again, this compound, phosphoenol pyruvate, is a high energy phosphate bond or energy rich phosphate compound. In the next and the final reaction of the pathway, here phosphoenol pyruvate is converted into pyruvate. Again, the enzyme pyruvate kinase, where this phosphate group is transferred to ADP to form ATP. Delta G of hydrolysis of PEP is more negative than that of ATP, catalyzed by pyruvate kinase. This is an allosteric enzyme and is inhibited by ATP. It is a tetramer of two types, M for muscle and L for liver, and we will talk about its control towards the end. End of part 1.